Could you all find a pew? Because we better kick off soonish, or we won't be able to do the Meet the Speakers thing on time. That's what's happening after this, Meet the Speakers, organised by our postdoc and PhD reps. I might go there as well, so you can meet me if you'd like. I told you yesterday that the best bit of being chair president of the British Society for Developmental Biology is handing out prizes and awards, and we gave our Tickle Award out yesterday. I'm going to give out two tonight, two gorgeous medals. I've got them both here. I'd love them both. So what I'm going to do is sort of set the scene and tell you about these two medals and about the two winners, and then one of them is going to give a talk. The other, it's a bit like an Oscar thing. They come up, they do you 2 minutes, and then they do a tour of several universities, and I'll explain it as we go along. Could you let me have some screen action? So these are three of our medals as of a little while ago, and we've got a fourth now. And the fourth one is in honour of Lewis, who died last year and was, you know, a pillar of developmental biology, one of the grandfathers of... He vivaed my PhD, and much of the time during the viva, he wasn't really focused because a package was coming that he was longing to receive. So my PhD was actually rather an easy thing. But anyway, he's lovely, and not only an amazing scientist, but a fantastic raconteur, communicator, advocate of developmental biology. And so we decided, we've been thinking on the BSDB committee for a while, that we would like a medal that honours, you know, the people who teach and communicate and sell developmental biology for us. And we commissioned Steph Nowatarski, who's a planarian postdoc in the States and also a fantastic artist, to come up with a design, and this is the design, and it looks even better now as a cast medal. And last year we awarded the first of these to Jamie Davis, who again just did the 2-minute Oscar receipt thing. I have to say I'm slightly worried about presenting Oscar-y type things. I was going to mock Rob Insall for his ridiculously fancy face mask, but I was worried that Laura Macheski would come up and slap me. So So I won't tease him about that. So here's, this year, I was involved in, I was one of the people who nominated this guy for the Wolpert Medal. And in part, I selfishly nominated him because one of my new PhD students, Jake, who Helen Weavers and I share running, he was a mathematician and he needed to learn fly stuff so that he could do his own wet lab experiments to do the maths on. And so Helen and I sent him on this Manchester fly faculty training program. And I joined in a bit because I don't know much about fly stuff either. And it's a fantastic course. But there's a lot more to it, to the sort of fly Manchester outreach thing. So, you know, some of the funders have these glossy brochures selling EDI outreach. This is EDI outreach. It's what Andreas does. These are all Drosophilists, you know, and that's where you need to get them. And that's where Andreas gets them. And he's infected the whole of Manchester and other places with this urge to engage folk. He also, when he was on our committee, this is sort of another selfish reason why he should get the prize, we've got the best archive of any society, and it's all down to Andreas. And he's an advocate of developmental biology and science communication. In fact, he even writes about it. So I've got your medal, Andreas. Please come up and don't slap me, and instead receive this medal.
Thanks so much, Paul, and thanks to the committee. It really makes my day, my year. And it's really beautiful. You're not exaggerating. So I'll have it in my rucksack, and if you want to see it, I show it to everybody who wants. So now I think it's really, really important that societies reward and recognize science communication as a service to science, to the community, which is fun, but also, of course, involves a lot of commitment, a lot of time investment, and very often we don't see then actually the recognition from our home institutions. And therefore, I think if there are other ways to reward this can only help. And I mean, how important it is, I've seen myself in Manchester when we merged the Faculty of Medicine and Life Sciences. Actually, we had one of the largest developmental biology groupings in the UK, and that was one that was crippled in the process. And if you think about it, we just fail to communicate the importance of our science. If one considers developmental biology as one of those disciplines that bridge between medicine and biology, because we are asking questions at the level at which medical symptoms become manifest, and we can provide satisfactory explanations for it and even applications. So this needs to be understood, and this is why it's so important that we all really engage in this. And if I think back of the discussion a while ago where the BSDB was trying to change its name, I think that was an alarming sign. And that was one of the reasons also why, when I was in the committee, I started with the advocacy work. And I would urge all of you, please look on the BSDB page. There is a tab on advocacy, which gives many, many arguments. And we invite everybody in our community to please help to further improve all these arguments, to enrich it further. Because it's so important that we all have our elevator pitches ready whenever we talk to politicians, our line managers, students on our courses, medics, journalists. So we should always be just prepared to be clear about why we do fundamental science, why model organisms are important, why developmental biology is important, why our projects are important. And I think by doing so, we can slowly move the goalpost. So, but that said, and I'm coming to an end now. That said, I must also say there is a lot to gain from science communication. I mean, it's on the one hand pleasure. On the other hand, it is something that makes you think about why I'm actually doing this research. And for me, I can only say it changed the way I think about my science. It changed the way I do my science. And it gave me fantastic statements for grant applications and papers. So there's something to be gained from it. And then, last not least, it gave me this wonderful medal. So I'm really grateful for this, and it's very much appreciated. So thank you very much.
I'm very relieved there were no tears or anything. Now, one thing I should say is no talk from Andreas, but the BSDB is funding 4 or 5 talks from Andreas at your various institutes. So if you fancy a bit of Andreas action, then please email Jens or I, who will help organise this. He can't go to everywhere, but we're going to fund 4 or 5 of them. Right, so next medal. Well, last medal of the evening. My AV guy is fantastic. Poor on sound. He really let me down yesterday, so I've got no movies with sound. But thanks. So this is our. We've had this medal for the longest. It's the Waddington. I have to say, it looks silvery there, but the one we've most recently cast is bronze stroke gold, because we didn't want Andreas's one to outdo the Waddington Medal. The Waddington Medal is our you know, the one that we give for a lifetime achievement, for someone totally kick-ass. And I was around on the committee when the first of these was nominated and awarded, and it went to Cheryl Tickle. So that's a really good start, and lovely to be receiving this medal. Now, it's standardly the case that we do a big reveal, and no one knows except the committee and me, who is going to get this medal and give us the Waddington talk. So I have to give you a few clues before we get to the big reveal. And I'm nervous about this next slide because it has a name on it that I fluffed up yesterday evening. Our principal nominator is Anahi Biguo Casas, which I hope is better than my yesterday's effort. And there were other nominators too, Austin and Jenny and James. Now, for some of you, that will give the game away, but for many of you, you still will have no clue who's going to receive this medal. So I'm going to show you a picture of our winner as a young'un. Now, Leonardo da Vinci designed a flying boat, and the parents of our winner tried to replicate that contraption. So the way it was supposed to work was that the operator rolled the paddles stroke fins with their chin. And eventually that would lead to this, and it's not clear whether this worked, but our winner's parents wanted to try and have a go at this. I'm going to show you our winner a little bit later, okay? This is in Edinburgh, the Centre for Genome Research, now called the Stem Cell Research Centre. So this person is a woman. She's a Scottish lass. She did her undergraduate degree in Glasgow and then in Cambridge, a PhD with Martin Evans, and then postdocs with Rosa Beddington. So you're starting, maybe some of you, to get a clue who it might be. It's this person, okay, next to a prepubescent Austin Smith. A year or two later, she is here at an EMBO Tübingen bash, and that's Rosa. Now, I did a PhD at ICRF Developmental Biology Unit in Oxford, and Rosa was along the corridor, and I did some experiments with her, and she is, or was, miraculous at grafting. And what I've heard is that our winner can graft on the long side, is as good as at graft, which is hard to believe, but the people I was told this are reliable folks. So she deserves the Waddington Medal just for her grafting skills. Okay. So this is who we're going to award the medal to, Val Wilson. She, as I said, is Scottish, and she's still in Scotland. And her website says that what she works on is trying to figure out why mice have long tails but not very, very, very long tails. And the answer is these cells, these green cells, which are neuromesodermal progenitor cells and can give rise to lots of different tissues. And I'm guessing, hoping, she's going to talk at least a bit about that in her talk. So the Waddington Medal goes to our hero, Val Wilson. This medal also is gorgeous. What do you think? But you need to move to your... You sure? That's you, isn't it?
Yeah, that's us. Great.
Sure.
Thank you very much. Well, thank you very much for that. That is... I have to say that I was completely stunned and have never been described as kick-ass before. So let's see. Maybe you'll be wishing that your medal, that this medal was delivered. The talk was delivered by a science communicator. I'll we'll see how we go on. Right, so I thought that in honour of the medal, I would give you the famous picture of Waddington's picture of how cell lineages diversify. So early, decades ago, he hypothesized that development occurs like in a sort of his schematic of embryo development is like a ball rolling down a hill. So at the top of the hill, the cell early in development can do everything, and as it goes through development, it makes successive lineage choices which restrict its options, so that, for example, a cell that is here maybe is making something like mesoderm, and a cell here making neural tube, and so by the time that it's made the neural tube, it can't make anything else, but it has other options within the neural tube. So this is a beautiful picture of development, and but I think that although we understand much more at the molecular level now how what exactly happens as cells make lineage choices, I think there's much that we still have to understand about what the hill is. So I'm going to tell you a little bit about the work that we've done over the years, and in trying to think about how to put together the work from long ago. I'm going to tell you work from really long ago and up till relatively recent. And as I was looking through my old data and photographs and things, I thought, actually, my picture of trying to finish a project is more like this. So instead of going down the hill, we're going up a hill. And in order to try and reach the goal, which is the finishing the project, publishing your results, my approach to trying to get to the top of the hill is often a lot more like this, which is this in Gotthard Pass, where I wander around, hopefully on my way upwards, but I sometimes find it hard to see the top of the hill from the false summits. So I was first thinking of this hill analogy at the end of my PhD, it turns out. So I looked at my PhD thesis and found this quote when I was trying to be erudite and quoting John Donne. So on a huge hill, cragged and steep, truth stands, and he that will reach her about must, and about must go, and what the hill's suddenness resists wins. So I was hoping to be to finally get to the truth. But this was what I wrote in my PhD, this is what I quoted in my PhD thesis, but further on in that poem, there was this: fool and wretch, at the last day, will it boot thee to say a Philip or a Gregory, a Harry or a Martin Evans taught thee this? So I know that I had this feeling that by the time that I was writing my PhD thesis, that I had made a lot of mistakes, but those mistakes I had to own, and they were my fault. And so sometimes, although we have plenty of support from our colleagues, sometimes only you can pull yourself up the mountain. So this is my friend and colleague, Alessandra Levigny. You can see that she's trying to pull herself up a mountain. So I'm going to begin the way that I often begin a lecture, a research talk, and then I'm going to break it and then talk with a slightly different slant. So what I usually do when I start a research talk is something like this. I'm interested in how the mouse head-to-tail axis forms. So this is a skeleton of a mouse, and you can see that it has a head, a neck, a trunk, and a tail. And you can see also these segments, the vertebrae, that are an indication of the segmented nature of the axis. So you can see that they're somewhat similar to each other. They're, each vertebra is a lump, but then it also, each vertebra also has its own unique character. So how does that mouse head to tail axis form? We're interested in how it begins and how it has a middle and how it has an end. So I often also show this slide here, which shows to scale mouse embryo from the start of, from just after implantation, so after it's just implanted in the mother's uterus, and getting bigger and bigger until about the middle of mouse gestation, which is about 10.5 days, and it's got another 9 or 10 days to go. You can see it expanding a bit as in these first few days, but then what happens is that it elongates. So you can see it starting to elongate here, and from then on it's getting longer and longer and longer. So this is when the axis is starting to elongate, and it does this over the period from around early day 8 until embryonic day 13. So the stages that I'll ask you to pay attention to tonight are these two stages, E8 and E10. So at E8, when it's formed, just the beginnings of these first two segments, the segments are somites at this stage, and the somites are the precursors of the vertebrae that I showed you in the skeleton, and of the segmented myotome, the muscles, the axial muscles. So you can see this E8 embryo has just a few somites, 2 or 3, and by E10 it has about 30 to 35, and in total the mouse will make about 60. So we also know that this elongation process happens because of a progenitor population that sits at the posterior end of the embryo. So in the early embryo it's called the primitive streak, and in the later embryo it's called the tail bud. So, final bit of introduction and background for you before we launch into the way that I wanted to arrange the talk. I'm just going to show you the arrangement of the tissues in the axis that we're going to hear about tonight. So, this is an E8 embryo. Actually, it's slightly later than the 2 to 3 somite embryo that you just saw. It's got about 10 somites. But if we take a piece of this embryo from here, you see the somites that have formed, the segmented blocks. So these are shown in pink here. And they flank the neural tube. So the somites are mesodermal. The neural tube is ectodermal in origin. Over that is the surface ectoderm, and under it is the endoderm, and underneath the neural tube is the mesodermal notochord. So that, I hope, is most of what you need to know about the anatomy of the embryo to understand what happens next. At the posterior end of the embryo, where I said the progenitors are, is the structure of the primitive streak. So I said the primitive streak is a source of progenitors for the axis. So in general, we know that at gastrulation, cells cells that are going to make mesoderm are in the epiblast. They move towards, so the epiblast is a columnar epithelium. They move towards the midline, where the primitive streak is, and undergo epithelial to mesenchymal transition, and exit from the primitive streak as mesoderm. And as they move out from this progenitor region, you have to imagine them moving forward as a sort of sheet of cells. If they're in the epithelial layer, they'll go on to make the neural tube. If they're in the mesodermal layer, then they'll go on to segment as somites. Okay, so while thinking about trying to present this and thinking about this whole issue that I have of winding my way, trying to get up to the top of the hill and perhaps taking wrong turns and perhaps not knowing exactly where the summit is, I think that one of the things that I almost you can think of as a signpost is that sometimes a particular embryo sticks in my mind. And this may be a common experience, but I don't know if it is. So I'm going to call this talk "My Favorite Embryos," and I'll try and illustrate from a few embryos some things about how results have happened during my career. So when I started my postdoc, after finishing the huge hill, cragged and steep PhD thesis, I came to the lab of Rosa Waddington, and she asked me what I wanted to work on, and I said anything but gene targeting, because that was what I had been doing. So in fact, that's what I'd been unsuccessfully doing for all of my PhD. So along the way, I had learned how to inject ES cells into blastocysts. And so she set me to work to do some blastocyst injections. So she and her collaborator, Bernard Herrmann, had recently isolated ES cells which lack the brachyury gene. So brachyury is a mutation, a very old mutation in mouse that Bernard had recently characterized at the DNA level and shown that it was a deletion of a gene, brachyury. So Rosa made some, Rosa and colleagues made some ES cells from these mutant mice, and we were then wanting to find out whether the brachyury mutation is cell autonomous. At this point, we didn't know that it was a transcription factor. Later we did. So the phenotype of brachyury null mutants is like this compared to wild type. So we have a nice long axis in the wild type embryos, the long tail, and brachyury null embryos have a shortened axis. So the question was, when we do the chimera analysis, when we transfer these into a foster mouse, we dissect them around mid-gestation and find out, do they look more like the brachyury nulls, or do they look more like the wild type embryos? So is it cell autonomous or not? So the answer is my first favorite embryo. So here is the tail of a normal embryo, and this is what happens in the chimeric tails. So we saw that these tails were really odd. They had, some of them, sometimes they were divided. They would be a forked tail, often short, not exactly like the null mutation. So because at this point we did not have a visible marker for where the cells were, you can see how long ago this was. This is 1992. We had to use something called glucose phosphate isomerase chromatography. So we had to find out how much of an isoenzyme, one form or other of an isoenzyme there was. So we could tell a little bit that these were chimeric. We could tell that they had more brachyury mutant cells towards the posterior end than they had more anterior in the axis, but we didn't know very much else. So then we had to, of course, mark the cells. So they had to be heritably marked. So I made these ES cells lacking brachyury with a ubiquitous LAC-Z marker, and they subsequently added a rescue transgene that expressed brachyury under its own promoter. So we injected these cells into blastocysts and compared the mutant cells with those that had the rescue transgene. So here's my next set of favorite embryos. So here we're looking at E8. Are you seeing? Yeah, you're seeing the pointer. you're looking at the back end of an E8 embryo. So just imagine rotating this embryo so that you're seeing the midline, the primitive streak. So this is the posterior end of the embryo. This is what happens with the null embryos. They start to collect in the primitive streak. So this is all the posterior midline, although they're pretty well dotted in elsewhere in the embryo. By E10.5, the cells have really collected in sort of huge numbers towards the posterior end of the embryo. The opposite is true of the cells that have the rescue transgene. So these cells are perfectly good at clearing their way out of the primitive streak. So that, together with the fact that brachyury, we know now, is expressed in the primitive streak region and in the tail bud region at later stages, tells us that brachyury cell autonomously permits the exit from the primitive streak. Permits cells to exit from the primitive streak and tail bud during the formation of somatic mesoderm. So the fact that brachyury is expressed continuously throughout axis elongation and the fact also that we saw, first of all, a small collection of cells early in development, and then a greater collection of cells that seemed to be the same process that was happening more and more as time went on in axis development, made me start to wonder about axis elongation itself. Is it one continuous process? So one quite influential paper for me was this one from Eddie de Robertis' lab, Gaunt et al., and it described the histologically and by marking cell populations in the late gastrulation stage xenopus embryo that, this is the sort of bottom line here, the tail bud is not an undifferentiated blastema, as previously thought. So people thought that perhaps the tail bud just had undifferentiated multipotent cells that then sort of were extruded from the tail bud and differentiated as the tissues that I've told you about. But here, they found that if they marked cells in the gastrulation stage xenopus, they could trace that into specific regions of the xenopus tail bud. And that, along with some really nice histological analysis, which I think is the first use of the word chordoneural hinge, showed that there is a structure within the xenopus tail bud that was that had a relationship with the earlier gastrulation stage embryo. So, to try and find out if this was if a similar sort of processes were happening in mouse, then I made use of the fact that embryos can be cultured. So mouse embryos are not the best of the developmental biology model organisms for classical embryology. However, there is a stage at which you can do this. So you can culture mouse embryos from early-ish stages for around 48 hours to a reasonably good stage. And the stage that was most useful for us was this early somatogenesis E8 stage. And so at this stage, you can manipulate the progenitor region and look at the descendants in an E10, up to E10. So I started to play around with mapping the primitive streak to see if you can map into the tail bud regions from the progenitor region at E8. Can you go from the primitive streak to the tail bud and see these defined regions that Gaunt et al. was looking at? So one thing that I think was very striking and interesting for us was that when you drip dye onto the surface of an E8 embryo, so you remember that I told you that embryos cells at the primitive streak at the midline are undergoing epithelial to mesenchymal transition and making mesoderm. So if you put di onto the surface of the embryo, then as you maybe would expect, you see label in the mesoderm. So this is the label extends into this pre-Semitic and into the Semitic mesoderm. But maybe even more surprisingly, if you take the posterior end of the tail and culture that, which you can do, then if the posterior neuropore is open and you still have a bit of exposed descendant of the primitive streak, then you also can trace cells from the epiblast into the mesoderm. So all you're doing here is dripping cells onto the surface of the epiblast remnant in the tail bud, and you see descendants in mesoderm. So the problem, though, was that we were a little suspicious about whether di was the right lineage label to use, because there were times when we thought that the cells were transferring di from one cell to another. So we turned instead to trying to graft cells, and this I'm very proud of. This embryo I left for Rosa to look at, and I came back to a note which said, "This is great!" And I think it was possibly the only time that she ever said anything that I did was great. So this is a grafted embryo where I grafted cells from one primitive streak of a Laxed labeled embryo into another primitive streak. And it has what I think is a classically good incorporation pattern, where cells are dispersed well along the axis, and they seem to have incorporated and made a normal type of tissue. So Paul told you that we do grafting. I thought I would show you a video of grafting, and this is actually made by a PhD student, possibly the most enthusiastic PhD student I have ever met, possibly in the world. So Yali Huang is famous for saying, "This is so cool." Anyway, so I hope you think that what she did is cool. So this is actually just grafting a set of cells into an E7.5 embryo. So just to let you know, the size of this is about 500 microns from the tip of the epiblast to the end of the embryo. So that's 0.5 millimeter, and we do that by looking. So you need a dissection microscope. So you're going to see a huge pair of forceps coming into view, and that will give you an idea of how of the size. So here we go. There's the embryo getting held by forceps, and there's a poor embryo getting spiked. So we put the needle into the embryo, and then we draw it out carefully while we're expelling the graft. And I hope that you actually saw that the graft is now there. So this is essentially what we do to try and get to look at how cells are behaving, either in their normal situation, so you graft from one region to the same region in another embryo, or you try and move them from one place to another. Okay, so I think two of my PhD students have really enjoyed doing the grafting. Actually, I enjoyed doing the grafting for many of my other PhD students, and it gives me a reason to feel wanted. So this is one of the grafters. Noemi gives me a reason to talk about this set of favourite embryos. So here, what we were trying to do was map the cells in the primitive streak. So we're using grafting from a GFP-labelled, ubiquitously expressing GFP embryo strain into the primitive streak of a non-GFP embryo. And so we started to try and see where do the cells in the primitive streak end up in both the differentiated parts of the axis and in the tail bud? So are any of them retained in the tail bud, and if so, where? So what we found, in particular, I'm not going to tell you all of this, but I'm going to highlight this one for you, that grafting cells at the border between the node and the primitive streak, that we call the node streak border, very, not very originally. So cells at the node streak border give rise to both somites and neurectoderm. So there's the neural tube, and there's cells in the neural tube, and cells in the mesoderm. Also, they give rise to cells in the tail bud, and this part of the tail bud I'm going to draw to your attention, because this is called the chordoneural hinge that I mentioned earlier, the junction between the neural tube and the end of the notochord. Okay, so we did quite a few of these mapping experiments and found that there were a few regions within the primitive streak and the tail bud that could give rise to both neurectoderm and mesoderm. So these are the neural and mesoderm fated regions. The node streak border that I've shown you, and this region here, the caudal lateral epiblast. So these two regions could give rise to both neurectoderm and mesoderm. Cells in the node streak border gave rise to cells in the chordoneural hinge, as I showed you. Cells in the caudal lateral epiblast, not so much. So our conclusion here, axis elongation in mouse, like other vertebrates, including Xenopus, involves continuous production of distinct tissues from localized sub-regions of the primitive streak and the tail bud. And a small region of primitive streak and tail bud give rise to both neural and mesodermal cells. Now, I'm going to take a little bit of a diversion here and show you some beautiful skeletons and say that there is underlying similarity in the vertebrate axis. So although there is astonishing diversity across the vertebrates, there's differences in both the sizes, the relative proportions of vertebrae, and also the numbers of vertebrae. There is always this repeating vertebral segments, and it turns out in the tail bud itself, there is also an underlying similarity, so underlying similar topology. So here is our old friend Brachyury, now as an antibody stain in purple, and it's expressed in the tail bud, and in fact all the regions of the tail bud, and include also in the notochord. And it's, we're also showing the expression of a neuro-specific gene, SOX2, and we see that where there's an overlap between SOX2 and T, in each of the organisms in which you can actually do do any classical embryology, that there's that this is marking a set of cells that seem to be able to go to both neurectoderm and mesoderm. Okay, so are these neural and mesodermal progenitors one cell type or several cell types? The grafting experiments that we did are usually composed of about 100 cells. So there's about maybe 100 to 200 cells. And so obviously this is not clonal, and so we can't tell that one cell is giving rise to two tissues. However, we had some clues. So this was us working our way up to some clues about whether these are one progenitor type or more than one progenitor type. So the first thing was that heterotopic grafts, where we take, so I've shown you node streak border into node streak border, and that's here. If we take the node streak border and move it into the rostral node, we see that the cells change fate. We move it into just beside it into the primitive streak, the cells change fate again to something much more like the cells, a homotopic graft of cells from either rostral node to rostral node or streak 1 to streak 1. So that suggests that there's quite a bit of plasticity of cells in the node-streak border. In the caudolateral epiblast, Philip Wymersk, another hero of very intensive experiments, helped me with any of the, or I helped him with the graphs, basically. I did the graphs, he did all the work. We grafted the caudolateral epiblast into the node-streak border. And here's my next set of favourite embryos. Actually, they weren't my favourites when I saw them at first, because they were really confusing us. Because when we grafted caudolateral epiblast into the node-streak border, we would get really widely different results. So sometimes we get embryos that contributed only to neural, and some of them were only to mesoderm. And we spent a lot of time trying to find out if there were regions of the caudolateral epiblast that were neural restricted versus mesoderm restricted. Turned out that that was not the case. In fact, it was exactly where we grafted. So just to show you the scale, this is 50 microns. So if we move these cells 50 microns back, then we see if we graft them into the node-streak border right at the front part of the node-streak border, they make neurectoderm and mesoderm. You graft them in the middle, it's kind of balanced, and you graft them just to the posterior end of the node-streak border, and it's mostly mesodermal. So these caudolateral epiblast cells will differentiate towards neural or mesodermal fates based on a very, very small change in their environment. And it doesn't matter where they came from in the caudolateral epiblast. They'll do what their graft site tells them to do. So, neuromesodermal fated regions change fate readily in response to a changed environment. And so that, again, suggests that we have cell plasticity. But, of course, we're developmental biologists. We can't show that we don't know this yet because we haven't grafted single cells. So Noemi and I tried very, very hard to graft single cells, and with no success at all. We always lysed the cell in the tip of the needle, and I was so gratified to see that recently Tatiana Solovyeva in Claudia Stern's lab has achieved it in Czech. She managed to graft single cells into the node and found that the cells can have multiple fates. So she's managed where we never did, and has found that Czech cells can have multiple fates. So not being able to graft single cells and show that they can have multiple fates, we're still asking this question: are these axial progenitors able to give rise to neurectoderm and mesoderm? The other question that is important, or that we found important, was: are these axial progenitors like stem cells? Are they stem cells, or are they something different? So this was really guided by a paper from Jean-François Nicolas, which really inspired me while I was working with Rosa. So here, I'll tell you a little bit more about his method, because we used something similar, but his conclusion was that the myotome, which I've told you, remember that the myotome is a part of the somite, so it's the muscle part of the somite. So this segmented myotome has mainly been produced by a unique, spatially organized pool of self-renewing stem cells that accompanies the formation of the anterior to posterior axis. So they found that these clones that were present in the myotome, if they looked like a stem cell-derived clone, they were always bilateral. And if they're bilateral, that means that they originated at the midline, and the midline, the last place that a myotome progenitor was at the midline, would have been in the primitive streak in the tail bud. So this was in our minds as we were thinking of how could we, is there really a stem cell within the tail bud? So this leads me to my next favorite embryo. So this embryo is that I've called the third generation embryo. I'll tell you how we made it. So we were trying to find out what happens to the cells that, so I showed you that we do these homotopic grafts, and we were looking at the cells in the tail bud and saying, are there cells in the tail bud? So then we wanted to know, are the cells in the tail bud stem cells? So because it's GFP, we can graft them again. So we grafted them into a second generation embryo. So here's the first generation. Embryos are grown to E10.5, and we grafted them back into the primitive streak, grow them again to E10.5. That gives us this type of embryo, and grow them back, graft them back again, and you get this third generation embryo. So this embryo has labeled cells in neurectoderm, mesoderm, and the tail bud. In fact, the caudoneural hinge of the tail bud, and it only works with the caudoneural hinge. So the conclusion is that a subpopulation of cells in the primitive streak region, the nose streak border and caudal lateral ephiblast, have neural and mesodermal potential. And these cells at the nose streak border additionally remain in the progenitor region over long periods of axis elongation and can recolonize the same axial regions on regrafting. So we're getting towards, do we have a stem cell? So I just want to bookmark here that we have not solved the heterochronic grafting. So I guess, I'm not sure if Lara Busby is in the audience, but we had she is working on grafting heterochronically, grafting from old to young, and looking at whether cells are able to give rise to or are able to incorporate well into the host. And here was a surprise for us that I have not yet solved, and is still one of my embryos that sits in my mind. Grafting from young into old gives us this. We don't, in contrast to this beautiful sort of salt and pepper incorporation, grafting from young to old doesn't incorporate very well at all. So I think we haven't really, I don't exactly know how to explain that. So are they, so then we want to know, are these progenitor stem cells? Was it one neuromesodermal stem cell type or several? So I've shown you that you can regraft cells from the tail bud into the primitive streak, and that they can give rise to the same somite several times. But still, we're stuck with the problem that we're grafting groups of cells. So to answer the question about can single cells do this, then we needed to turn to a clonal analysis. And what we did was turn to Jean-François Nicolas and the system that he had invented. So this uses a LAC-Z form of LAC-Z, this LAC-Z gene, which contains a duplication which renders most of beta-galactosidase out of frame. And only on a spontaneous intragenic recombination, which is done by the cell's own repair mechanisms, but rarely and at random do you repair LAC-Z. And that happens, of course, in the cell that does it and all of its descendants. So if you dissect an embryo at a stage after this clone is initiated, you'll see the cells that the clone has contributed to. And if you dissect enough of these, then in theory, you could reconstruct the entire developmental history through piling up the clones that you've generated. This is one example of a clone that labels notochord. We dissected perhaps several thousands of clones. We haven't reconstructed the entire developmental history, but we have done something. So this took about 10 years from start to finish. But right at the very start came another of our favorite embryos. This embryo was one of the first embryos that we dissected, and it has labeling in the neurectoderm and the mesoderm, the somites, and the caudoneural hinge. So this embryo never occurred again in that transgenic strain of mouse because the frequency of recombination was too rare, and so we had to reconstruct another mouse which had a more frequent recombination. And finally, we got a bunch of clones that told us that there is indeed a dual-fated neuromesodermal progenitor. And these cells act like stem cells in the sense that they can be retained in the primitive streak and the tail bud and continuously produce the neurotube and the somites. So this you may be able to see is not telling us exactly where that neuromesodermal progenitor is. I think that using our previous history of doing grafting and mapping tells us where it would have to be. It would have had to be in the caudolateral epiblast and the node streak border, and then in the chordoneural hinge. But more recently, Charlène Guillot, who was working with Olivier Poquier, has actually filmed cells with neural and mesodermal fates. So, and they did indeed come from the anterior caudolateral epiblast. Okay, so just taking this sort of a bit full circle and a little bit more up to date. More recently, Carolina Gibantif, I don't know, who was working with John Marioni and Bertie Gutgens, kindly invited me to come and have a look at their embryos, which were a reanalysis of brachyury null chimeras. And if you're not colorblind, then you'll be able to see the collection of cells in the primitive streak, and they then did a transcriptomic analysis of the brachyury mutant cells within the wild-type embryos. And very sort of pleasingly, I think, they found that when they put these brachyury mutant cells into wild-type embryos, then neuromesodermal progenitors are overrepresented in those chimeras and not in control wild-type chimeras. And underrepresented are the somites. So they also were able to show that there are several developmental trajectories of cells into the anterior somites, and the brachyury mutation acts at this point to stop cells going into posterior somites and diverts them into neuromesodermal progenitors, which are then stuck in the primitive streak and are then carried along with tail development. Now, how much do you feel like you need a beer, and how much do you feel like, because I can probably skip very nearly to the end, and I'm going to get to a conclusion. There was a lot about pluripotency, but I think I'm going to leave that for another day. Instead, I'm going to try and give you a little bit of conclusion about what in our system we think the ball rolling down the hill is. So in the primitive streak and the tail bud, and especially in neuromesodermal progenitors, we think that a combination of intracellular and extracellular cues are moving cells from one state to another, from an early state to a mid-state, and so on, until the end of axis elongation. And part of that involves, of course, for those of you that that know anything about axis elongation, would of course think, does it involve Hox genes? Of course, that must be the case. So we think that the progressive opening of the Hox cluster plus the inputs from different WNTs and the internal feedbacks within the cell are moving the cells from early to late until they're eliminated at the end of axis elongation. So we're nearly done. I wanted to tell you about one more thing, and that was that there are some twists and turns that in my career that didn't really lead to the top of, well, maybe. We did publish something from this. In fact, it led to a really nice excursion. I spent several years trying to generate a bunch of gene trap mutants. One of which taken over by Simon Bullock when he was a PhD student. So it shows how very old I am. I was the postdoc in day-to-day charge of Simon. What this mutation was in a heparin sulfate sulfotransferase, and it was, I think it was nice because it meant that I was able to experience a completely other field, the field of heparin sulfates. So these are extracellular molecules that are cofactors for signaling, and this was one of the first mouse mutations that showed that there were developmental effects of modifying or creating different modifications of heparin sulfate. However, the rest of the gene trapping has lain unpublished and heroic yet unpublished work in folders within my cupboards for years. And so I felt like this was really giving us the importance of climbing the right mountain, unlike what actually happened here, which was an excursion with some of my friends and colleagues, where we went up to Glencoe, and I took everyone up the wrong mountain. And you can see the sheer drop here and the mist rolling in. And there was actually quite a hairy moment. We did get down the other side, and nobody was injured by the odd ankle. But this, I think, is also, I think although we need to get to the top of the hill, but we also need to enjoy the moment, just as Anestis and Morris are doing here, having a good smoke at the top of the mountain. So this, now I'm into the acknowledgements. So first I want to say thank you very much to the developmental biology group at the ISCR and in CRM where I moved. So this features some well-known faces. I think you can see Josh here. It's Josh Brickman. There's Sally, our very own Sally Lowell. And so I want to say thank you to them all for inspiration, constructive criticism, and lots of outings to real-life mountains. And here are the lab in various iterations. And I want to say thank you ever so much to everybody. And yeah, you can see lots of honours students have come through the lab, and many of them have done a characterization of some of the clones, and lots of collaborators. And a big thank you, especially to the Medical Research Council, who've funded a lot of my work over the years. And I think I'm going to leave it at one more thank you to the mice, and Rosa's drawing of a mouse. What I do now, do I go away?
No, stay with a second. On this occasion, and every time the Waddington's awarded, there are no questions. But do you not agree it was totally appropriate to describe Val as kick-ass? It was a fantastic talk. We're very proud to have given you that medal. Have you lost it already? No.
Thank you.
Don't lose it. Now, if you are thinking that you know the next Andreas Wolpert Medal or the next Val Waddington Medal, then you need to tell Jens or me or anyone on the committee, because this has to be passed on to someone equally kick-ass next year. Right? Though you get to keep that one. Now you get to meet the speakers, and I've forgotten where that is. Can you shout it out a little louder? The Fusion Bar in Warwick is where you can get to meet the speakers. Off you go. Have a good evening.